Hello, my name is Su Lwin. I'm a dermatology registrar and honorary clinical research fellow at St John's Institute of Dermatology at King's College London in the UK, and I'd like to present to you the safety and early efficacy outcomes of this first-in-man clinical trial using lentiviral fibroblast gene therapy to treat 4 adults with recessive dystrophic epidermolysis bullosa, called the Lenticle F trial. So this is a collaborative study between King's College London, Guy's and St Thomas's hospitals and our colleagues at Great Ormond Street Hospital. So RDEB, first of all, it's a really severe blistering skin disease affecting the skin and the mucous membranes. And in this disease, patients suffer from chronic wounds and erosions and lifelong pain. Eventually, they succumb to skin cancer, cutaneous squamous cell carcinoma by the age of 40. There are no effective therapies for these patients currently, and therefore, with the hope of developing new therapies, we look into gene therapy. Now, in terms of the pathology, type 7 collagen protein, which is the main constituent of anchoring fibrils, these are fan-shaped structures that ensure the top layer of the skin, the epidermis, to be adhered to the bottom layer of the skin, the dermis. In RDEB, this is either deficient, absent, or malformed, and this is due to loss of function mutations in COL7A1 gene. So what we have done is, with our colleagues at Great Ormond Street, we developed a self-inactivating lentiviral platform. This is to say that the infectious components of the vector has been deleted so that it doesn't keep replicating after transducing into the cells. Now, using this, we have previously shown in mice that this is an efficacious, potentially efficacious therapy. Now, what we have done is conducted a phase 1 clinical trial and treated 4 adult patients with RDEB. So figure 1 here illustrates the vector platform in figure 1b, and in figure 1a it illustrates what happens in the Lenticle F trial. So we took a skin biopsy sample from the RDEB patients, took it to the GMP lab at Great Ormond Street, isolated the fibroblast cells, transduced them with full copy of the COL7A1 gene, and then inject them back into the intact skin of these patients in order to look at safety and efficacy. So just moving down slightly here, so the objectives as mentioned are safety and efficacy, and looking at specifically in terms of efficacy whether or not the type 7 collagen has been restored and the morphology of the anchoring fibrils, as well as the evidence of any of the transgene, i.e. the full copy of the COL7A1 gene that we inserted back is actually present in the injected skin sites. The trial took place at Guy's Hospital, and the gene therapy was manufactured at Great Ormond Street Hospital. It was a 12-month-long follow-up in this trial. So the subjects' baseline characteristics, there were 5 subjects, so to speak. The reason was because the subject 1 was enrolled twice due to the failure of delivery in the first time round. There were in total 2 males and 2 females, so the first male was enrolled twice. And also they had different subtypes of EB ranging from generalized severe, meaning very severe end, to milder form such as the inversa. We successfully manufactured the gene-modified fibroblasts and then injected them back into the left upper arm over 1 small centimeter squared, and then analyzed the skin biopsy as well as bloods. So going on to the outcomes, as you can see here, the subject 1, 2, subject 3, 4, and then 5 being the most severe patient. So this figure 5 demonstrates the collagen 7 expression back in the skin after injection, and you can see here in this box and whiskers plot that 50% of the injected skin site had significant increase in expression of collagen 7. However, there was no evidence of new mature anchoring fibrils formation. So moving on to figure 7 here, we then looked at whether or not the actual transgene is still present after 12 months, and we did indeed find the presence of transgene itself only in the injected skin and not in the uninjected skin, of course. And this is the summary to conclude that in this study we demonstrated the safety, and fortunately none of the patients developed any severe adverse reactions, and only mild adverse reactions related to the procedure and not the gene therapy itself. And what we have done is provided the foundation to build for further dose escalation studies with larger sample size and larger cell doses in the future. Thank you.